What do light, sleep, and memory all have in common? The answer is, of course, the brain. Now, the memory and sleep portion are obvious, but what do lights have to do with the brain? Well, one of the most useful methods we have to study the brain is the ability to isolate specific brain cells called neurons and either activate or deactivate them. You see, brain cells, neurons, talk to each other by, you guessed it, activation or deactivation. When they activate, they will send an electrical or chemical signal to their neighbors, and when they deactivate, they will not. So, in short, this method allows us to use light to control communication in the brain. Okay, sounds cool, but what can we do with it? The answer, of course, is we can turn on and turn off memories. Allow me to explain. Let's assume that you're driving to work, and there's construction, and you have to take a new route than you usually do. And on the first day, you have to pay very close attention to the turns, the stop signs, the stoplights, other people driving, etc. Now, by the third or fourth day, you're getting pretty good at this. It's because you remembered your past experience. And you're able to do this because there are specific neurons that map onto this experience. And by map on, I mean they activate while you're doing this task. And while you're asleep, these cells will reactivate over and over and over again, basically kind of playing the scene over and over again to solidify the memory. However, what would you think would happen if I deprived you of sleep by talking to you about neuroscience? Well, the obvious answer is these cells would not activate and you would forget. It's a long established principle in neuroscience that sleep is important for memory. And this happens by sleep allowing these cells to reactivate and then allowing the memory to solidify. No sleep, no reactivation, no memory. However, this raises an interesting question with the tool we mentioned earlier. What would happen if we allowed someone to sleep, but we artificially prevented their neurons from firing, or that is, from activating? This is the question that I explored with Dr. Brittany Clausen during the fall of 2019. Now, one's first instinct is to go get some humans and then let them sleep and then mess around with their brains. Well, there's a lot of problems with this. The main issue is that no one usually volunteers their brain to scientists for research, so we have to find a way around this. And one of the most popular animals used in neuroscience research is the mouse. And an obvious problem here is we cannot talk to mice, so how do we know if they remember something? Well, we can use something that is called a fear-based memory task. Allow me to explain. You take a mouse and you put them inside of a box, and then, let's say, you shock the mice very lightly, and then the next day you put them back in the box. Chances are they're going to remember that, hey, I was shocked in this box, so I'm going to be afraid of it. And usually a mouse will communicate its fear by freezing, by literally trying to stand still as much as it can. Now, if we were to put a mouse inside of a box and just show them an image, we wouldn't really expect much of a response. Even if we let them sleep, put them back in the box, and show them the image again, they're probably going to be bored, if anything. However, if we put them inside the box and we both shock them and show them the image, then they're likely to remember that experience. And then let's say the next day we put them in there and we just show them the image. The fact that we showed them at the same time the previous day, the mouse is going to associate the image with the shock and now become afraid of the image. They were once not afraid of the image, but now they are. This allows us to see if a mouse has remembered something. In this case, they're remembering that the image is associated with a shock. We first gathered two groups of mice. One was going to be our control and one was going to be our experimental. In short, one was just going to be normal, we weren't going to mess them in any way, and the other, we were going to deactivate their cells. So, the first group, we put each mouse inside the box, shock them, show them the image, let them sleep, and then the next day, put them back in the box, and we measured how much they froze. And as you would expect, for the control group, they responded as you would think. They were afraid of the appropriate image. However, for the other group, we did the same task, put them in the box, shocked them, showed them the image, etc. But while they were asleep, we shined some special lights on their brain cells that prevented them them activating, just a certain part, not the entire brain. So, in short, these cells were not activating, though they were sleeping. So, what do you think happened? An obvious hypothesis is, oh, they probably don't remember anything. Well, here's where it gets interesting. This group of mice were very confused. We put them back in the box, we showed them an image, and they were afraid of it. That is, the image that we paired with the shock. Well, that can't be right. We deactivated their cells, and they should remember. Well, this is where science gets interesting. Oftentimes, we try to predict what's going to happen, and then we don't get the results we expect. In this case, we learned that, oh, they were afraid of every single image we showed them, not just the image associated with the shock. And then we concluded that, oh, we were just knocking out the visual portion of the memory and not the fear. So the mouse knew to be afraid, but did not know what to be afraid of. This illustrates 2 points. One, that memories are multifaceted, and oftentimes that different parts of the brain are going to control for different aspects of a memory. And something even grander is that science does not always turn out the way we expect it. Oftentimes it doesn't. But that's the beauty of science. It oftentimes leaves us with more questions than answers.